Imagine trying to unravel a really complex medical mystery, piece by painstaking piece, until suddenly a hidden connection clicks into place. Well, that's pretty much what we're doing today. We're diving into a really groundbreaking case report. It gives us the very first in vivo evidence connecting a rare genetic condition, multisystem proteinopathy 1, or MSP1,
Right.
which is caused by mutations in the VCP gene, to Parkinsonism, and crucially, to a specific protein biomarker. It's quite a significant step forward in understanding how these conditions actually work.
What's really fascinating here, I think, is how this specific report bridges a gap that's been there for a while in our knowledge. For years, our insights into this VCP-related Parkinsonism, they mostly came from postmortem studies, looking at tissue after someone had passed away. But this research, it moves us into the realm of live, verifiable evidence. It gives us this unprecedented window into the active disease processes happening inside living people. So this isn't just finding a correlation, it's a direct observable finding. And that could really change things for early detection and maybe even for targeted treatments down the line.
Okay, so to really understand this breakthrough, let's back up a bit and clarify what we're talking about with MSP1. So, multisystem proteinopathy 1, it's a rare genetic disorder. It's autosomal dominant, adult onset. Autosomal dominant just means you only need one copy of the altered VCP gene to get the disorder.
That's right.
It used to be called IBMPFD, which stood for inclusion body myopathy, Paget's disease of bone, and frontotemporal dementia. Quite a mouthful.
A real mouthful, yeah.
But those were its sort of signature symptoms, the cardinal features. The name now, MSP1, with multisystem in it, really drives home how wide-ranging its effects are on the body.
Absolutely. Yeah. And at the heart of MSP1 is this VCP gene. It provides the instructions for making the valicin-containing protein. Now, this protein, it's everywhere in the body. It's ubiquitously expressed. It's part of what's called the AAA plus protein family. They're a diverse group known for using energy, ATP, to kind of remodel things in the cell.
Right.
You can think of the VCP protein as maybe a master regulator of, let's call it cellular proteostasis, basically keeping proteins in good shape and getting rid of damaged ones. It's essential for keeping cells healthy. It's really involved in two major pathways for getting rid of old or damaged proteins: the ubiquitin proteasome system, or UPS, and the autophagy lysosomal pathway, the ALP. Now, when you get these pathogenic VCP variants, these mutations, they mess up these systems. It's like a breakdown in the cell's quality control. And that leads to misfolded proteins building up. Things like ubiquitin, TDP43, alpha-synuclein, even tau aggregates. And this buildup, this cellular trash, can become toxic and cause widespread problems.
Okay, so it's like the cell's waste management system is broken.
That's a great analogy. Yeah, gets clogged up.
But despite this really broad impact at the cellular level, Parkinsonism showing up as a symptom of MSP1 is, well, surprisingly uncommon. It only happens in about 3 to 4% of cases.
That's correct. It's quite rare. And the VCP-related disorders themselves, they have this incredibly wide range of symptoms, well beyond those classic IBM PFD features. You can see forms of ALS, a type of Charcot-Marie-Tooth disease, hereditary spastic paraplegia, various muscle diseases, myopathies that look a bit like muscular dystrophies.
Wow.
So this diversity, it really makes diagnosing and understanding these rarer presentations like the Parkinsonism a real challenge.
Yeah, and that challenge kind of brings us back to a key question, doesn't it?
It does.
Without that direct in vivo evidence, how can we reliably diagnose these complex processes while the patient is still alive, to intervene earlier or more effectively?
Exactly. As you said, postmortem studies had previously shown this alpha-synuclein pathology in people who had VCP-related Parkinsonism.
Right. And alpha-synuclein, or alsin. Many listeners will know this is a key player in Parkinson's disease, especially when it misfolds and clumps together inside neurons.
Precisely. But not having that live evidence, seeing that aggregation happening in a living person, that's been a huge hurdle in definitively connecting the VCP mutation to the synucleinopathy.
And that lack of live evidence has really been the sticking point, hasn't it? Which leads us perfectly to the patient described in this new report. This study follows a 76-year-old man. His really complex symptoms ultimately helped find that missing piece. So 5 years before the study, he started showing signs of a Parkinsonian syndrome. Initially, they thought it was PSP-like progressive supranuclear palsy because he had significant problems with balance, postural instability, and kept falling. But his problems actually went back much further. He'd had this profound, long-standing muscle weakness, especially in the limbs closer to his body, the proximal muscles, since he was a young adult.
Making everyday things difficult.
Yeah, like just getting up from a chair or climbing stairs, things we take for granted. It was really debilitating for him for a long time, and his condition just kept progressing. By age 71, he developed that forward bent posture, a tremor when holding positions, then a resting tremor, classic Parkinson's sign, in his right hand showed up 3 years later. By 75, his balance was really bad, marked postural instability, and he developed foot drop on the right side. This led to frequent backward falls, and he needed a walker.
And the family noticed other changes too.
Yes, exactly. His family reported changes like apathy, sort of a lack of motivation, and social withdrawal. But what really clued the doctors into a possible genetic link was his family history. His mother, his daughter, and his granddaughter all had scapular winging where the shoulder blade sticks out abnormally.
Ah, that's a strong clue. That pattern really points towards something inherited. So the diagnostic workup they did for him was incredibly thorough, and it uncovered this whole collection of findings that, taken together, really suggested something more complex than typical Parkinson's, something genetic. The neurological exam, it confirmed that severe widespread muscle wasting, the very noticeable bilateral scapular winging, significant proximal weakness. He even had a positive Gowers sign.
Which indicates weakness around the hips and pelvis.
Exactly. Pelvic girdle weakness. On top of that, he had the resting tremor, rigidity, that plastic-like stiffness, slowness of movement, bradykinesia, which was worse on his left side, and also slow eye movements, saccades, and that severe postural instability. Quite a picture.
Wow, yeah, a lot going on.
A lot. And the other test added more pieces. Brain MRI showed some subtle shrinkage in the frontal cortex, but nothing dramatic, which is actually pretty typical for VCP-related Parkinsonism.
Yeah.
But a DAT scan SPECT, which looks at dopamine transporter levels in the brain, that clearly showed reduced signal bilaterally in the nigrostriatal pathway.
Okay, so that confirms the dopamine system dysfunction seen in Parkinsonism.
Precisely. Confirmed nigrostriatal dysfunction. Then, looking at the muscles, EMG showed a widespread myopathic pattern. Muscle MRI revealed significant fatty replacement in his thigh muscles, quite asymmetric too, and a muscle biopsy confirmed dystrophic changes.
So the muscle disease was clearly present.
Clearly. And then the bone aspect. Blood tests showed high levels of bone alkaline phosphatase and beta cross-lapse, markers of bone turnover. And a whole-body bone scan confirmed polyostotic Paget's disease, Paget's affecting multiple bones.
Okay, and cognition.
Cognitive tests showed mild global impairment, but particularly affecting executive functions, things like planning, problem solving. So you see it's hitting multiple systems: muscle, bone, brain motor control, cognition.
Right, so after this really extensive workup, uncovering this very distinctive mix, the myopathy, the Parkinsonism, those pyramidal signs, Paget's disease, what was the final piece? This unique pattern just screamed genetic cause, didn't it? And next-generation sequencing delivered the answer. It found a pathogenic heterozygous mutation in the VCP gene, specifically the C.277CT PJAR93C variant.
That genetic finding tied it all together, explained this complex multi-system picture.
And then came the real clincher.
Yes, and here's the pivotal moment, the aha, that this whole deep dive is really about. They performed a skin biopsy.
A skin biopsy, okay.
Yeah. And remarkably, it showed abnormal deposits inside the nerves of the skin. Deposits of phosphorylated alpha-synuclein.
Wow, in the skin.
In the skin nerves. This is the first time this specific protein buildup has been demonstrated in vivo in a living patient with VCP-related MSP1 and Parkinsonism. Finding this biomarker, which we know is so relevant to synucleinopathies like Parkinson's, in an accessible tissue like skin, instead of needing brain tissue after death. Well, that's incredibly groundbreaking.
That really is something. It potentially opens up a whole new way to diagnose or monitor the condition.
Potentially, yes. It's a highly sensitive and specific biomarker in this context.
And just one more clinical point on this patient treatment. He was given high-dose carbidopa-levodopa, the standard Parkinson's drug. But he didn't really respond well. He had poor responsiveness. And the study notes this is seen in some, though not all, cases of VCP-related Parkinsonism.
Yes, that variability in treatment response is another interesting feature. It hints that the underlying mechanisms might be slightly different from typical idiopathic Parkinson's disease.
So why? Why does this one VCP mutation cause this cascade of problems? Let's dig into the cellular mechanisms a bit more.
Okay, so as we touched on, these pathogenic VCP variants impair the cell's ability to clear out damaged proteins. The proteostasis machinery gets jammed up. This directly leads to the buildup of misfolded proteins, and importantly here, that includes alpha-synuclein. There are even experimental models showing that inhibiting VCP can actually promote the seeding of alpha-synuclein in neurons, basically making it easier for those toxic clumps to form and spread.
Okay, so the faulty VCP directly contributes to the alpha-synuclein problem.
Directly, yes. But there's another critical piece. VCP's role in mitochondrial health.
Ah, mitochondria, the powerhouses of the cell.
Exactly. And impaired mitochondrial function, problems with their dynamics. These are expected consequences when VCP isn't working properly. And we know, of course, that mitochondrial dysfunction is a major, well-established contributor to neurodegeneration in Parkinson's disease itself.
Right.
What's really intriguing, though, is that VCP actually interacts directly with PNK1 and Parkin2 genes, whose mutations are known causes of familial Parkinson's disease.
Oh, interesting connection.
It is. PNK1 and Parkin are crucial for mitophagy, which is the process of clearing out damaged mitochondria. So this suggests there might be a shared pathway. A single VCP mutation could be disrupting both protein clearance and mitochondrial quality control, hitting processes that are central to Parkinson's pathology. That could explain the Parkinsonism symptoms we see in these VCP patients.
Okay, let me try and summarize that. So we're looking at a situation where these VCP mutations basically trigger a kind of perfect storm inside the cell.
That's a good way to put it.
You've got the protein waste disposal system getting jammed, leading to that dangerous buildup of alpha-synuclein. And you've got problems with mitochondria messing with the cell's energy and health. And these two issues seem to converge, maybe even make each other worse, driving the Parkinsonism symptoms in these patients. It's not just one pathway failing, it's like a domino effect.
Precisely. A cascade failure affecting multiple critical systems.
Zooming back out to the clinic, it's clear there's a lot of variability in how VCP-related Parkinsonism presents. We mentioned the age of onset can range hugely, from 30 up to 71 years old. And the response to dopamine treatment varies too. Some people do well, others, like the man in this case study, don't respond much at all.
Right.
And sometimes it can even look like other atypical Parkinsonian syndromes, like corticobasal syndrome, with its own specific set of motor and cognitive issues.
That variability is key. And alongside our case study, it really seems to reinforce the idea that the alpha-synuclein finding might be quite specific. The evidence points towards alpha-synuclein deposition being mainly restricted to those VCP mutation carriers who actually develop Parkinsonism.
Okay.
Remember that postmortem study we mentioned earlier, Spina and colleagues from 2013? They looked at brains from VCP patients. 2 patients who had both frontotemporal dementia and Parkinsonism did indeed show Lewy bodies and Lewy neurites, the classic alpha-synuclein pathology.
Right.
But crucially, a third patient in that same study who had the VCP mutation but didn't have Parkinsonism showed no alpha-synuclein pathology.
Ah, I see. So the pathology tracked with the symptoms.
It seems so. And our current in vivo finding from the skin biopsy strongly supports this specificity. It suggests it's not just having any VCP mutation, but perhaps specific mutations or context linked to Parkinsonism that trigger this particular alpha-synuclein problem.
Which naturally leads to the big question.
Exactly. Why? Why does only a small fraction, that 3–4%, of people with VCP mutations end up developing Parkinsonism? And mechanistically, how do these specific mutations actually drive the aggregation of alpha-synuclein? These are really the big unknowns, the frontier of our knowledge right now.
Do we have any hypotheses, any ideas floating around?
Well, there are possibilities. It could be that different VCP mutations affect cellular pathways in slightly different ways, leading to different clinical outcomes. Some might disrupt protein clearance more, others might hit mitochondria harder, perhaps. Another possibility is genetic modifiers, other genes in a person's background that might influence how the VCP mutation manifests. Or maybe environmental factors play a role too, interacting with the genetic predisposition. We just don't know for sure yet.
Lots more research needed there.
Okay.
But bringing it back to the practical side for listeners, what this means now is that clinical guidelines are starting to reflect these deeper insights. They now recommend considering VCP genetic testing in certain cases of Parkinson's or Parkinsonism, especially if the onset is unusually young, if there are atypical features that don't quite fit the standard Parkinson's picture, if there's a strong family history suggestive of a dominant inheritance pattern, or, importantly, if the person also has other signs of MSP, like that significant muscle weakness or Paget's disease of bone.
Right. It helps put the whole clinical picture together.
Yeah. This deep dive really underscores why that genetic testing can be so valuable for getting an accurate diagnosis, and maybe, looking ahead, for guiding potential future treatments tailored to the underlying VCP dysfunction. So, wrapping this up, what this deep dive has really illuminated is how this in vivo skin biopsy finding is such a significant step. It establishes that direct, detectable link between VCP mutations, the synucleinopathy involving alpha-synuclein, and Parkinsonism, a link previously just inferred from postmortem studies. It's a huge leap, isn't it? From historical observation to finding evidence in a living patient that could be used for diagnosis. It definitely moves us closer to really understanding, and hopefully one day treating, these incredibly complex neurodegenerative diseases.
Absolutely. And if you think about the bigger picture, this case really highlights how one single gene mutation can disrupt multiple absolutely critical cellular processes. Everything from protein quality control and waste management right through to the health of the mitochondria.
Hmm.
And all these disruptions converge to cause complex conditions like Parkinsonism. It really makes you wonder, doesn't it? How many other rare genetic disorders might have similar, currently hidden underlying mechanisms just waiting for that crucial first in vivo observation like this skin biopsy to bring them into the light.
Yeah.
The journey of discovery for many of these conditions is truly just getting started, and I think this case provides a really powerful example of how we can move forward.
That's a fantastic point, a powerful thought to leave everyone with these intricate, hidden biological connections that scientists are slowly, carefully unearthing, one piece at a time. Keep asking questions, keep exploring, and definitely keep diving deep with us.